Hello my dear students and welcome back to my channel. In today's video, we will be discussing about the topic protein fingerprinting or peptide mapping. This is also known as the two-dimensional analysis of protein. So without wasting any time, let us quickly move towards the video. So, the topic for today's video is protein fingerprinting or peptide mapping. This is a very important topic from the chapter number 2 of class 12 biotechnology and is very important because almost every year questions comes from this topic in your board exams. So now let us come to the topic. This technique was first of all developed by the scientist V.M. Ingram in 1957 when he was studying the protein sickle cell hemoglobin. The principle of this technique is that every protein has a unique peptide map which makes it different from other proteins and thus serves as a unique fingerprint for that particular protein. Means that like DNA fingerprinting, every individual's DNA is unique, and when cut with an enzyme, the pattern of fragments obtained for that particular individual is different from other individual. In the same way, every protein has a unique fingerprint. Means if two proteins are cut with same enzyme, then the pattern of fragments obtained from both the proteins will be different. This technique is also known as 2D analysis of protein. Here 2D means two-dimensional analysis of protein. Why? Because two techniques are applied here. The one is paper electrophoresis and the second is paper chromatography. In the two-dimensional analysis, one technique is applied firstly and then that technique is applied at 90-degree angle to the second one. This technique is mostly used in the study of sickle cell anemia. Now, what is sickle cell anemia? We all know that sickle cell anemia is a genetic disorder where the hemoglobin becomes sickle celled, and because its shape changes, so the oxygen carrying capacity of the RBCs is also hampered. Therefore, the disease is known as sickle cell anemia. Like we all know that normally the RBCs are biconcave in shape, but the sickle cell hemoglobin becomes sickle cell shaped. Sickle means the moon. Uh, as you can see, the figure has been drawn. Half moon shape becomes. So, as you can see, there is a difference in the shape of the RBC, and therefore the hemoglobin content of the RBC will be hampered, and thereby the oxygen carrying capacity of the sickle cell RBCs will be hampered, and therefore anemia will occur. Sickle cell anemia is also known as a molecular disease because it arises due to substitution of amino acid valine in place of glutamic acid at the sixth position in the beta chain of hemoglobin. So, now let us see how this protein fingerprinting or peptide mapping is done. So, this is generally a comparative study, and here we will compare the sickle cell hemoglobin with the normal hemoglobin. So for this, first of all, we will isolate normal RBCs on one side and sickle cell RBCs on other side. From the normal RBCs, normal hemoglobin will be purified, and from the sickle cell RBCs, the sickle cell hemoglobin will be purified. After purifying both the hemoglobins, they will be digested with the enzyme trypsin. Now, trypsin is a proteolytic enzyme. Proteolytic enzyme means that this trypsin will digest protein segments or cut protein segments. So, how it will cut the protein segments? There are particular sites where this trypsin enzyme will act. It will not act randomly and cut the peptides in whichever manner it wants, but it will cut the bonds adjacent to the lysine and arginine amino acids. Means wherever a lysine residue is found, it will cut the adjacent peptide bond, and wherever the arginine residue is found, it will cut the adjacent peptide bond, and therefore the peptides will be released. Now, after the peptides are released, paper electrophoresis will be done at pH 2.0. This paper electrophoresis will be done on the Whatman filter paper, where the samples will be placed on the middle of the paper, and therefore the peptides will move towards their respective electrodes upon application of the electric field. After this paper electrophoresis, the sheet of this paper electrophoresis is dried and then attached to the paper chromatographic sheet at right angles to it, and the second dimension of the technique, which is the paper chromatography, is done with the butanol, water and acetic acid as a solvent. Now, in paper chromatography, the principle which is used is the partition coefficient of the peptides, where the more hydrophobic peptides will move longer to the paper. Means the peptides which are more hydrophobic will move away from the solvent system. Now, after this paper chromatography is done, the peptides will be visualized by using ninhydrin, and the peptides will occur as yellow spots. Now, this chromatogram is compared of both the normal and the sickle cell RBCs, and after comparing, it was found that one of the peptides was different in the sickle cell RBCs. Means out of the four spots which we got on the chromatogram, one spot was differently placed in the sickle cell RBC. It means that all the three peptides which were found similar, they would be similar. So now we will do the peptide sequencing of the different peptide which was found on the chromatogram. And after the peptide sequencing of this differential peptide, we found out that there was a substitution of the valine amino acid in place of glutamic acid in the sixth position of the beta chain of the hemoglobin, and therefore this single substitution of amino acid resulted in the drastic change in the shape of the RBCs, where its shape changed from the biconcave to the sickle cell shaped. So this was all about the protein fingerprinting or peptide mapping. Hope you liked the video. If you have any queries, do write in the comment section below. And if you are new to my channel, please subscribe to my channel. Thank you all for watching.